The second major type of sex determination that we're going to talk about is genic sex determination, and we're not going to spend too much time on this. This is a type of determination system that we see in some plants as well as in a lot of fungi, protozoa, and some fish species. So, genic sex determination is where sex chromosomes are actually not even present. Okay, so genotypes at one or more loci on autosomes are actually determining the sex of the individual. So, again, genic sex determination is not being determined by sex chromosomes, it is actually being determined by genotypes at one or more loci on autosomes. All right, so as we progress through sex determination, just make sure that you have a really good handle on the difference between chromosomal and genic sex determination. Keep in mind, chromosomal determination is there is actually going to be gender specific chromosomes, and those chromosomes are going to be different between the two gender species. So males are going to typically be one genotype, and females are going to be a second genotype. And then genic determination systems is where the autosomes are actually going to determine the sex of the individual. So let's talk a little bit about environmental sex determination as our third major system. So this example is best visualized with slipper limpets, and these are actually larvae that settle on a piece of open space, basically. So it could be wood species like on a pier or rocks, but the larvae are going to settle in this unoccupied space, and they're going to develop into a female. And so, as they are developing into the female species, they're actually going to start producing proteins and chemicals that begin to attract more larvae. Those larvae are then going to move in and set on top of the original larva, and eventually they're going to stack up on one another. And what ends up happening is the males on the top actually have the ability to switch sex, developing into females. So this takes a little bit of time, but just realize that it is an environmental type of determination system. Okay, so as this in this system is really what we call sequential hermaphrodism. And this is where each individual organism or limpet can be both male and female, but it just doesn't happen at the same time. So the sex ends up being determined by the position within the stack of limpets. The fourth type of determination system that we're going to talk about is very specific to Drosophila, or the common housefly. So just to give you a little bit of background, Drosophila has a total of 8 chromosomes, and those chromosomes really split out into 4 pairs. So 3 pairs of chromosomes are going to be autosomes, and 1 pair is going to be sex chromosomes. The female is going to have the genotype of XX, and the male will be XY, but it's very different than the human system. Y actually does not determine the degree of maleness in Drosophila species. So you'll see what we're talking about in just a few minutes. So the way that they actually came up with this hypothesis was they worked through the scientific method to make these conclusions. Their original hypothesis was that the fly's sex was actually determined by a balance between the genes on the autosomes and the genes on the X chromosome. So the X chromosome would contain genes that had female effects. So they would code for protein that were very specific to the female phenotype. And then they hypothesized again that the autosomes would actually contain genes that would code for proteins specific to male effects. And so what the result of those two hypotheses was that the fly sex was actually predicted by the X chromosome to autosome ratio. So X to A is the X chromosome to autosome ratio. So more current research had actually determined that genes on the X chromosome are the sex determinant. Okay, so they influence the autosomal chromosomes a little bit, and so the effect of those autosomal chromosomes are very indirect when compared to the genes that are being expressed on the X chromosome. So what ends up happening is there's a control of expressional timing. Okay, so there's going to be a very specific time when the X chromosome genes are actually going to be expressed and upregulated to go through transcription and translation. So as we take a further look at this genic balance system in Drosophila, there is the prediction that that the number of X chromosomes is going to be if we take the number of X chromosomes and divide by the number of haploid sets of autosomal chromosomes, then we get the sex of the Drosophila or of the housefly. So if we have an X to A ratio of 1% or 1.0, then that individual will be female. However, if we have an X to A ratio of 0.5, then that individual will be male. So as you can see in this figure here, we've got our female fly. She is going to produce these sets of autosomes, and so set number 1 of autosomes, set number 2, and set number 3, and then she's also going to produce 1 set of sex chromosomes. Okay, so in our male species, they're also going to produce 3 sets of autosomes, but the sex chromosome is going to have a very different Y chromosome compared to the X. All right, so I encourage you, if you're having trouble following the Drosophila logic, to look at Table 4.2 and actually read this section in more in depth in your book. And this will actually help you be able to determine what the sexual phenotype of an individual fly is based on the ratio of their sex chromosomes to their autosomes. All right, and finally, the last sex determination system that we're going to talk about is in humans and also in most mammals. So we see this in most of our livestock species as well. And so the sex determination system in humans really goes back to the Y chromosome determining maleness. And more specifically, there's a gene on the Y chromosome called the SRY gene. And this is really going to make the difference and actually result in the male phenotype. So the SRY gene is going to be very close to the telomeres on the short arm of the Y chromosome. And it's going to go through expression, through transcription and translation, and if it is present. So if it's not present, like we have 2 X chromosomes, therefore the SRY protein or the SRY gene is not present, then we're not going to get any protein material being upregulated. Therefore, by default, that individual will be female. Okay, so if we have no SRY protein being produced after transcription and translation, then by default the ovaries will develop. There's not going to be any anti-Müllerian hormone or AMH produced. Therefore, the female reproductive tract begins to develop. So we'll get the paramesophenic ducts as well as the oviducts, the uterus, the cervix, and the vagina. And so eventually that XX genotype will completely turn into the female tract. However, the opposite of that is if we have an X and a Y chromosome present in an offspring, that individual is going to be male. And because the Y chromosome contains the SRY protein, that SRY protein is going to be produced after that gene's gone through transcription and translation. And in the presence of that protein, the testes will actually begin to develop. If the SRY protein is not present, keep in mind the ovaries are going to develop. Okay, so with the male, if that protein is present, those testes are going to develop. That's going to result in Sertoli cells and their production of anti-Müllerian hormone and dihydrotestosterone. And both of those are going to start um, being secreted in a really pretty quick pulsatile fashion, and those are going to end up being um, cleaved into the testosterone chemical that we know and dihydrotestosterone as well. And the testosterone chemical is going to allow the development of the male duct system to be produced, and dihydrotestosterone is then going to develop the secondary sex characteristics such as the penis, the scrotum, and the accessory sex glands. So again, whether we get males or females directly goes back to the presence of the SRY protein that only shows up on the Y chromosome. All right, so as we continue to study sex determination in humans, it's important to keep in mind what exactly the role of sex hormones is. So first of all, the X chromosome is going to have genes that are important to both sexes, male and female, so you at least have to have one copy of the X chromosome present. The male determining gene is located on the Y chromosome. So if an individual is born with one Y chromosome, then SRY protein will be produced, and that individual will develop into a male. If there isn't a Y chromosome produced, then that SRY protein will not be produced. Therefore, a female will develop by default. And keep in mind that the fertility associated with humans is actually on the X chromosome. So fertility in females is only going to be exist if two X chromosomes are actually present.